Welcome to Derms on Drugs, a video podcast brought to you by Scholars in Medicine, the best educational platform in dermatology, and totally free. Derms on Drugs is where cutting-edge derm meets hit or miss comedy. I'm Matt Cyrus, and each week I'm joined by my residency buddies, Dr. Laura Ferris and Dr. Tim Patton, where we use our 60 years of combined derm experience to discuss, debate, and dissect the hottest topics in dermatology. It's everything you need to know to be on the cutting edge of derm, and it'll be the most fun you've had while actually learning something useful. New episodes drop every Friday on Scholars in Medicine, Apple Podcasts, Spotify, and any other somewhat major podcast platform where you might get them. All right, so let's go ahead and jump into our special post-AAD episode where we are going to get into our favorite late-breaking research, and I'm going to kick it over to Dr. Ferris to get us off the ground.
All right, so I am going to talk about the iconic LEAD study. So this was the first release of results for the phase 3 study of the targeted oral peptide that blocks the IL-23 receptor called icotrokinra, which has something that kind of means IL-23 in Greek. So this was
All Greek to me.
It was a phase 3 study. So I have talked before about the phase 2 study. I do kind of like this study. So I promise this might be maybe my last time talking about it for a while. But this is the phase 3. Icotrokinra 200 milligrams once a day, which was not in the earlier phase studies, versus placebo. So primary endpoint at 16 weeks. Then patients who were on placebo did cross over to the icotrokinra. So this was adolescents and adults, but mostly adults in the study. So they actually presented week 16 and week 24 data. So these patients were kind of typical like psoriasis clinical trial patients, BSA around 25%. About a third of them were bio-experienced. So what were the top line results? PASI 90 at week 16 was 50% of patients. Interestingly, at week 24, that went up to 65%. PASI 100 was 27% at week 16, but 40% at week 24. IGA 01 was 65%, and then it was 74%. So kind of similar to what you saw in the phase 2, but maybe a little bit slower to get to those results. They also had scalp-specific IGA, so 0 or 1 clear or almost clear. 80% of patients who came in with scalp disease were clear or almost clear at week 24. Nothing new safety-wise. So, I think this is interesting. I think it's going to be interesting to see how this works in special sites, psoriatic arthritis, all kinds of stuff. It was kind of bold to go forward with the dose that they had not looked at in phase 2, but it does seem like it worked as well as BID dosing, which I think is going to be important for an oral drug.
Do you think the results will keep getting better when we go out to 36 weeks? And at this point, what existing drug would you say it's most, if I, like, because I keep in my head, like, here's how good I think of it. What would you think it's most at the moment, just efficacy-wise?
Yes, I know we talked about the phase 2 data, and the closest I came was Cosentyx, but maybe this data you would say this looks like Cosentyx, but slower. But it's once a day oral drug.
Yep. Okay. All right. Looks good to me. All right, Patton, go ahead.
All right. Efficacy and safety of oral deucravacitinib in patients with cutaneous manifestations of lupus erythematosus results from PAISLEY CLE, a global phase 2 randomized double-blind placebo-controlled trial. Data was presented by Victoria Worth. I didn't even know that there was a study in 2022 that looked at Deucra and SLE. That shows you how much I'm in the literature. I'm involved. They had some skin cores in that study, but this study, the PAISLEY CLE study, was specifically looking at cutaneous lupus. Patients could have systemic, fewer than 50% of the patients did in this particular study. Adults were randomized in a 1-to-1-to-1 fashion. Placebo, Deucra 3 milligrams BID and Deucra 6 milligrams BID. There was a crossover from placebo at week 16. So the primary endpoint was mean percent score change from the baseline in CLASI A at week 16. And for placebo, the mean percentage change was 28.4%. And for the two Deucra groups, it was better, 47 and 50 for the 3 milligram and the 6 milligram, respectively. Secondary endpoints were mean improvement from baseline and then CLASI 50. And they were also better for Deucra. CLASI 70 was a post hoc analysis, and that was better for the two Deucra groups. The CLASI 70 numbers were odd. The placebo group, like 15.9% reached CLASI 70. The lower dose Deucra, 3 milligrams BID, it was 49.5. So 15.9 for placebo, 49.5 for Deucra, 3 milligrams BID. That's a huge difference. The higher dose Deucra percentage was lower. It was 29.5. It was a lot. It was like 20% difference. And the p-value was 0.273, I guess, not statistically significant. So that 70 data was weird, but overall, it looks like Deucra could be a good option for CLE patients. I included a little, there was actually a review about small molecule biologics in lupus, and it was like a meta-analysis, and Deucra like came out on top. And I mean, granted, meta-analysis and all the things that go along with that. But so Deucra, yeah,
it's
maybe.
it's hard for me to get that. Like, Deucra is a pretty effective drug, but like if you're going to study something that's expensive, why not study the JAK inhibitor? That's going to be. But I get it. I guess I get it. I
Better,
don't know.
maybe better, more acceptable safety. I mean, it does make sense. It hits the interferon alpha pathway, right? So it totally makes sense for lupus. And looking at,
Yeah.
like, I was at that late breaker, like the pictures are pretty impressive.
Yeah.
Like, sometimes you look at lupus studies, you're like, I guess that worked, but you're like, oh, that looked better.
I guess that's true. I have no sense of what CLASI 50 looks like, right? You have a sense in your head what PASI 90 looks like or EASI 75, but yeah, CLASI we're not used to any endpoints in. Meh. Okay.
Yeah.
All right, we're going to move on to my first article. This was QY201 in adults with moderate to severe atopic dermatitis, 12-week results from a dose-finding phase 2 trial. So here's what was interesting. This is a JAK1 TYK2 dual inhibitor. So this seems that it would be the equivalent of giving somebody both Sabinco and Sotyktu. So, right, Sabinco's highly JAK1 selective, and obviously, so TYK2 is a TYK2 inhibitor. So 200 patients, 1-to-1 to 1-to-1, 50 people in each arm. And what was impressive, this had by far the best results I've ever seen for any study. So if these results are reproducible in phase 3, their highest dose, 20 milligrams BID, got over 80% of people to EASI 75 in 12 weeks. Normal data, so like high dose Rinvoq gets about 70% of people there. So this is meaningfully better than high dose Rinvoq. They got about 70% of people to IGA 0/1. Again, in Rinvoq, that number is more in the 50 to 60 range. So this would be if these numbers were real, it'll turn out to be the most effective thing we've got. And it makes me think maybe some of my super challenging patients, maybe the key thing to do is have them on Sabinco, which is a pure JAK1 inhibitor, and give them some Sotyktu samples on top of it or something. But we'll see. But that was just most impressive data I've seen. Either you any comments?
No, I think it's going to be interesting to see, where these drugs end up, and who knows, maybe that'll also work in things like lupus. I think we're just seeing the tip of the iceberg on what these combinations can do. It'll be interesting.
It's interesting just that we still haven't really seen a whole lot of progress in atopic derm efficacy from when Dupi came out. It's just interesting how different it is from psoriasis.
Yeah.
All right,
and usually things work better in phase 2 than phase 3. So we'll see how it does in phase 3.
True.
Yeah.
All right, Dr. Ferris, let's go on. You give us our TYK2 inhibitor update.
Okay, the TYK2, the molecule of the podcast. So I'm going to just quickly go through 3. So first one, efficacy and safety of ESK-001, a highly selective oral TYK2 inhibitor, moderate to severe plaque psoriasis, phase 2 results presented by Andy Blauvelt. And so basically the dose that they looked at was, was 40 milligrams BID. Sort of the pharmacokinetics behind that was that while TYK2, has partial inhibition of TYK2 activity throughout a 24-hour period. Their claim is with this dose, 24-hour full inhibition of TYK2. So maybe it'll work better. So what did they see? PASI 75 at week 12 was 64%. They had an open label extension. So if we look at week 52 data, that PASI 75 response was 77.5%, PASI 90, 61%, PASI 100, 39%. So how is that? It's pretty good. If you look at to do the Matt Zyrus, how does it work like Sotyktu? PASI 75, similar for Sotyktu is 72%. For Sotyktu, PASI 90 is about 45% versus for this drug, it was about 61%. PASI 100 is not really published for Sotyktu. I think I've seen it presented and it's sort of, I'm going to guess it's like around the 20% level. So don't quote me on that. So about double that PASI 100 of 39%. So, kind of interesting that, there's a lot of people working on TYK2 inhibitors. So there was another one. It was Inventus Bio from China, D2570. So patients randomized at, 3 different doses. And basically, if you look at PASI 75 in that group, it was like up to 90% versus placebo was 12%.
Wow.
If you look at PASI 100, they were 39 to 50%. PASI 90, 70 to 77%. So also looking pretty good. And then finally,
it's hard to understand. So if the idea is that the one right before this, the ESK drug, is above the whatever threshold for 24 hours, how the hell can another drug be meaningfully more effective?
Yeah, it's hard to know. I mean, I don't know if it's hitting. One could argue one might be less selective, one might be more selective. I think they're all allosteric inhibitors, but yeah, bioavailability. I mean, who knows? Like those are generally in vitro things. So that was kind of interesting. And then the last one was InnoCare Pharma, which is also Chinese, ICP 488. And so they had, PASI 75 results that were like 78%. Their PASI 90s were like 35 to 50%. PASI 100 was more like 11%. So I would say this is a little bit more like Sotyktu in efficacy. So lots of, development around TYK2. We'll see which one wins.
Yeah, that's it. So it'll be an interesting race. Hopefully it's not a race to the bottom. Hopefully it's a race to the finish line for TYK2.
It's a lot of energy.
A lot of energy.
A lot of investment.
Yes. All right, Dr. Patton, let's go on and do the Dupy for BP.
Yeah, efficacy and safety of dupilumab in patients with bullous pemphigoid results from Liberty BP Adept Phase 2-3 study data presented by Frederick Kaw. I'm not sure if you pronounce that X. He was French. He was very French. The design of this trial was published in Advances in Therapy in March 24. And so that's kind of what I would show during the slide. It's a double-blind, placebo-controlled 52-week study of Dupy plus steroids. I think it's very important to mention that it was Dupy combined with steroids, dosed in a very specific and well-controlled way. In adults with moderate to severe BP, primary endpoint was proportion of patients achieving sustained remission at week 36. It was a tough endpoint, right? Patients had to be in complete remission, so no active disease at week 36, no steroids after the initial taper at week 16, no relapse once steroids were discontinued, no rescue therapy through week 36. They had to meet all of those endpoint, and it was reached by about 20% of patients on Dupy versus 4 on placebo. So that doesn't seem like a huge number, but I think that primary endpoint was challenging.
Were they using any topical steroids?
What?
Do you know if they were using any topical steroids?
I don't.
Okay.
I want to say it was probably not allowed, but I don't know that for sure.
Yeah, probably not, but I couldn't find it anywhere I looked.
Yeah. There were other endpoints greater than 90% improvement in BP-DI, peak pruritus scores that the Dupy group did better at. Cumulative prednisone dose was mentioned. It was kind of interesting. In the Dupy group, it was 1,678 milligrams lower over 36 weeks. That works out to 6.6 milligrams a day, if I did my math right. It doesn't seem like that much of a difference, but maybe over 36 weeks, that would be a huge difference. I don't know.
What was the average? Do you know what the average prednisone dose was for the people who were not on it? Like, how much of a reduction was that?
Yeah, they did present that, but it wasn't in the little abstract. And I'm not one of those persons that takes a picture of every slide when they put up the thing.
Not a nerd like Ferris.
I love that. My
It's
camera roll is full.
it's very odd looking. I don't know. It's dystopian to me. Everyone's.
I have that confidence. We're good.
So I think Dupi will help physicians manage BP patients. But when you have BP patients, you're like, steroids will get this under control. We know this. How do we get them off steroids? These are old people. They have comorbidities. If 80% of the patients are still on steroids, did you really kind of get you to your main goal? And with rituximab out there, which may allow us to do it better, I don't know. But Dupi's easier to administer. Side effect profile is nice. I think more people will use it. It'll be interesting to see where Dupi fits in for the treatment of BP. I don't know. I think it will help us manage these patients, but yeah.
But did they put them on like a high enough dose to clear and then say, now let's taper? Or did you have to, they did do that. They did come to totally clear on steroids.
Right. So 6 weeks of corticosteroids, like at a moderate dose to shut BP down. I can tell you from seeing a bunch of BP patients that have been managed by other dermatologists, nobody does that. Nobody keeps their patients on 30 or 40 milligrams of prednisone for 6 weeks. That was how the study was done. And then they tapered it more rapid than I think what you would have done in clinical practice. But I think they had to see if Dupi worked. You know what I mean?
So maybe there's room for this, like with slower tapers and.
Well, do we have any idea how doxycycline and minocycline work in BP? Because that would be the obvious combo: is put them on that and Dupi, and it's all very safe and blah blah blah.
Anti-inflammatory matrix metalloprotein 9 inhibition by dox. I mean, I don't think we know.
Okay, fair. Fair. So do you think this will be like seeing the I don't know if they had pictures of the thing or whatever, but they're going to show you the best pictures. So who the heck knows so far? Have you used much Dupi off label in pemphigoid?
A decent amount. It's plus minus. I mean, some people absolutely respond, and other patients, they come back and they're like, I stopped that shot. It wasn't doing anything. Like, I still have all these blisters and
It'll be the Tesla of BP.
Or maybe they'll find biomarkers. So in the urticaria space, it looks like it's going to be definable. Like, this will be a good Dupi patient. This won't. That data should be coming out pretty soon.
Yeah.
All right, we're going to jump onto mine, which I love this idea. So 3-phase adaptive clinical trial to evaluate the human factors of patient-led patch testing protocol for the diagnosis of allergic contact dermatitis. So this is one of the most timely, interesting things I think I've seen somebody really try and do. So the basic idea here is they have this thing called the patient journey, which is like the patient, I don't know what they're doing, answering questions, something, and it helps individualize allergen selection. And then rather than putting like 80 or 100 or 120 patch test allergens on somebody's back, you put 20 on both, 10 on each of their volar forearms. And then they developed an app that the patient then, after they take them off in 2 days, 2 days later, they do this AI deep learning visual thing to interpret the patch test spots. And so, and I will tell you, literally nowadays with CAMP 2.0, the Contact Allergy Management Program for the American Contact Derm Society, and AI, between those two things, most patients, once they figure out what they're allergic to, can probably figure it out themselves. And this, if you're just testing people to 20 allergens, you could literally be like, hey, put this app on your phone, do the stuff for the next month, then come back. It'll tell us what to test you to. We'll put the tests on and go on your way. You don't have to come back again. It could make it so much more patient friendly. Now, you're not going to get into like occupational cases and stuff like that, but for routine patch testing, this could be a really big breakthrough. And I did go back and look because as a patch tester, you always talk about, well, you want to put it on their back because that's where we developed all the allergens for, and your skin's different in different places. There have been a few studies looking at that, and it not a ton, but a few, like literally 2 or 3. And it does look like putting allergens on your volar forearms is fine, that you don't get too many false positives or false negatives. So this is feasible. I'm interested to see where it goes. I don't think it's something you can go out and start doing tomorrow, but next year this could have developed to the point where we're able to do it.
So
That would be,
one question from this, because
yeah.
I sat through this and I was like, I thought it was kind of cool for the AI aspect, but like half of these patients had an allergy to propolis.
Yeah.
Yeah,
So
what's up with that?
that was with propolis, which for our listeners, propolis is a component of beeswax. It's basically theoretically processed out of the beeswax and whatever comes from the pollen, that kind of stuff. But you see a ton of propolis patch test reactions if you're patch testing full panel, and in my opinion, the vast majority of them are false positives. They are real positives, meaning there is clearly a reaction on your arm, and it clearly looks like allergic contact dermatitis. But I never saw clinical relevance to it. It can be a marker for fragrance allergy in theory, can be a marker for beeswax allergy, but I never really saw any clinical relevance to it, meaning I didn't see the people get better. But
Okay.
you're right on the money. You saw a lot of positives, but I think they were not truly indicative of allergic contact dermatitis.
All right.
All right, let's go on to our next one, Dr. Ferris.
All right. My last one that I wanted to talk about was totally different. This was laser assisted drug delivery to treat non-melanoma skin cancer. So I just thought this was cool. This was not like, and this was, sometimes like you get all these big pharma studies. I also like this was sort of a single center training program where they did this study and presented it. So biopsy proven basal and squamous cell carcinomas that were under 2 centimeters. They had 30 cancers in 23 patients. This was not a randomized study, but what they did was they had this dual wavelength, and I'm not a laser person, but non-ablative laser, which I kind of know what that means. And so they did like a little pulse with the laser, 1550 and 1927 nanometers, and then with a 1 to 1.5 centimeter margin. Then they put tirbanibulin, which is Klisyri, on first dose in the office. Then they had them do it for 4 more nights at home. And then they saw them back and like rebiopsied and reevaluated at 1/6 and 12 months. And so what was kind of cool was like there was a 100% response rate. These were, they're skin cancers that were lower risk. They've been shave biopsied. But they did and they showed pictures like the cosmetic outcome looked really good. And the moderator said, well, you should have done, laser alone, Klisyri alone, and then the two together. It should have been a 3-arm study. I totally get that, but kind of an intriguing idea that maybe there are ways that we can deliver drugs and we can do things more creatively to deliver, topical agents into skin cancers, whether it's laser or injection. And, I think deserves more work, and also kudos to people in training for doing clinical trials. It's not always easy, and being up there against, the big pharma companies.
Why the hell did they have to use a laser? Why couldn't they have just used like a
Pins?
or hyfricator and like zap, 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 zap. Okay, here's some terbinafine to put on there.
Yeah, I mean, I think that the idea is that kind of targets your like these little, like not like little holes, but not, not ablative, into like you get an epidermal and dermal penetration. So it sort of lets you
Okay.
open up and target drug into those areas. Yeah, could you
It
maybe
gets
get this
deeper.
another way, like the little smallpox vaccine things that they used to use? Like
Yeah.
maybe there's another way to do it,
There's
but
probably a bunch of that laying around.
we got a bunch exactly.
All
With
right.
I'm
measles
probably the
coming
only
back,
one here who
smallpox is
actually
probably
got
going to come
vaccinated. I'm
back.
the only one old enough to have gotten vaccinated against smallpox.
Yeah,
My
yeah.
mom said she tried so hard to find a doctor to give me the smallpox vaccine, but they weren't giving it anymore.
She was actually just trying to give you smallpox. That was,
She could tell early on.
yeah, exactly.
All right, Patton, what do you got?
All right. Also, skin cancer-y, PD-1 directed intratumoral immunotherapy for cutaneous carcinomas. Interim results from an ongoing study of Intasil PH762. Data presented by Mary Spellman. What is Intasil PH762? It's a proprietary compound by Phio Pharmaceuticals. It delivers a silencine. Is that how you say it? Silencine RNA. Silent.
Yes, I think so.
To cells through some sort of spontaneous delivery, which I guess is kind of a unique for siRNA technology. The self-delivering molecule enters the cell. In this case, it would be a T cell. Binds to an RNA silencing complex, RISC, which then goes and finds the messenger RNA that is going to make the cell make PD-1, and it destroys that messenger RNA so that T cells won't express PD-1. So it would serve the same function as a PD-1 blocker. Patients got 4 doses of intratumoral injection over 3 weeks and then underwent surgical excision 2 weeks later. 6 patients with SCC, 2 complete responses, 2 partial, one of which was like a 90%, and 2 non-responders. So for intratumoral stuff, is this good? Is this bad? I don't think we really know. Small study. 1 patient with metastatic melanoma had no response. So just a kind of a neat molecule, I guess. But, I don't think we have any big intratumoral data out of the PD-1s or anything like that to see how these numbers compare.
No, we just have
do
that.
have ongoing trials, and there's been some like early phase data for intralesional cemiplimab for basal cell carcinoma and squamous cell. So, I think that the response rate was better than this for intralesional cemiplimab, but.
And we got that TVEC and the intratumoral TVEC stuff coming.
Yep.
But yeah, I got a little stuck on one of the slides. I was reading it and I was like, because it shows the 2 strands of DNA, and then one is the guide strand. And I was like, guide, I've never seen what's guide. And then turns out it's just the guide strand.
Yeah. And it's RNA, but yeah.
Okay. Moving on to our last 2 here.
It's getting late in the podcast.
All right, so initial results from the Signal AA study, randomized placebo-controlled phase 2A trial of bempikibart.
Sure.
Bempikibart,
You got it.
Novel IL-7 TSLP bifunctional receptor antagonist in patients with severe alopecia areata. So basically, this is a monoclonal antibody that binds to one thing, but kind of like Stelara binds to one thing, but blocks 2. This binds to one thing, but blocks both IL-7 and TSLP. And basically, it turns off autoimmune T cells without having much effect on normal T cells, is the basic idea. So it does seem to have some element of blocking response to a totally new antigen, but it doesn't really immunosuppress you. You don't see people getting opportunistic infections. This is a very small study, 44 patients, kind of a proof of concept, and the SALT 20 scores were not great. But I can tell you that as an investigator, it was really hard to enroll this trial because the JAKs had just come out, so everybody with alopecia areata was going on JAKs. And what was really shocking was that they had a number of patients who continued to regrow, get better and better for 6 months after the end of the study. So this could be our first biologic for alopecia areata, and it could be something that really has a long-term effect of turning around this autoimmune process. So it's an interesting new mechanism of action. They're going to be pursuing it in a number of other things, including vitiligo. So 2 diseases we don't really have any biologics in yet, but just the long-term effect was what was coolest. And then the other study, similarly, the long-termness is what's important here. So this is rocatinlimab. This is an Amgen molecule. And so rocatinlimab significantly improved clinical signs and symptoms by targeting OX40 receptor positive T cells in patients with moderate to severe atopic dermatitis. Results from the phase 3 Rocket Horizon trial. And so this is a monoclonal antibody that targets the OX40 receptor and blocks OX40 and OX40 ligand from co-stimulating each other. reduces memory T cell populations. This was basically a normal atopic derm study, except the endpoint was 24 weeks instead of the usual 12 or 16. And the results were not like mind blowing. So Easy 75, they got about 33% of people at week 24. IJ01 versus placebo got 13%. IJ01, they got 19.3% versus 6.6 for placebo. Got to tell you, my experience on the trials, I thought the drug worked better than this in the trial. So, but the big takeaway is it had not plateaued at 24 weeks, and because of its depleting memory T cells, there is real belief that this could be long-term disease modifying, where you take this for 6 months or a year, and then you're able to be off therapy for long periods of time, perhaps indefinitely. So that's kind of the interesting thing. The other cool thing with this drug, or interesting thing, small number of patients, but when I say small, maybe 20%, will get fevers and chills after the first dose. And it's pretty impressive. Like whenever they come back in and tell you about it, they're like curled up in bed, like shaking and sweating, which I'm wondering if that's a marker for efficacy. If those people, have a better outcome, but nobody's cut the data that way yet. But so that's just an interesting new pathway that will be that's on the horizon for atopic derm. And again, the main benefit doesn't seem to be rapid efficacy. It seems to be potential for long-term remission. Is the big takeaway. I don't know. Either of you have any comments on either of these?
I think I thought the pickup art, or however you say it, study was super interesting, and it's TSLP. So you could imagine like it's maybe not just atopic, or, that could work in atopic derm. That could work in, it's interesting for AA. The safety looked good. And, again, like we talked about Jackson and AA, like 6 months is still kind of an early endpoint, right? So.
Yeah, no, I agree. And you're right. So this has both, because IL-7 is more in the Th1E pathway. So this has got kind of a Th1 and Th2 slant to it. And so it really is broad what it could work in. I think they're going after some pulmonary indications as well, maybe eosinophilic esophagitis, that kind of stuff.
Maybe BP?
Maybe BP. Maybe we'll finally get something.
All right.
Yeah.
All right. Well, that sums it up for the late breakers that we here at Derms on Drugs thought were the most interesting this year at the AAD. So thank you for joining us. If you've got questions, comments, ideas for topics we should cover on the show, shoot us an email, questions@dermsondrugs.com. Again, questions@dermsondrugs.com. And we hope you learned a few things. We hope to laugh once or twice. And mostly, we're hoping you're planning to join us next week. Until then, I'm Matt Zyrus.
I'm Tim Patton.
I'm Laura Ferris, and we are Derms on Drugs.